In this video, I will examine electron transport chains. What are they? How do they work? Why are they used? As their name indicates, these chains transport electrons. They do this with a series of oxidation-reduction reactions. As electrons move down a chain, their energy is used to pump protons across the membrane to form an electrochemical gradient. The gradient is then used for cellular operations, such as the generation of ATP by chemiosmosis. Let's start with oxidation-reduction reactions. Electrons can move from a donor molecule to an acceptor molecule. Since electrons are negatively charged, the electrical charge of an acceptor molecule is reduced. The donor becomes more positive. Early chemists thought that the loss of an electron involved oxygen, so they called the loss of electrons oxidation. Since reduction and oxidation happen simultaneously, we call them a redox reaction. Electron transport chains are redox reactions occurring one after another. Each reduced acceptor becomes in turn a donor, and the electron is passed down the chain from one molecule to the next. The process ends when the electron is passed to a final acceptor, which keeps the electron. A reduced final acceptor must be replaced before the process can continue. In cells, electron transport chains are more complicated because a membrane holds the acceptor-donor molecules near one another, and the energy of the electrons is used to pump hydrogen ions, which are protons, across the membrane. Prokaryotes pump protons out of the cell. Many different electron acceptors and donors are used in electron transport chains, such as flavoproteins, coenzymes called ubiquinones, and proteins that contain metal ions, such as cytochromes. Let's look at an electron transport chain more closely. This one is found in a bacterial cytoplasmic membrane and transports 2 electrons at a time. An electron carrier called NAD brings 2 electrons to the chain from the reactions of glycolysis and the Krebs cycle. Reduced NAD attracts 2 hydrogen ions, so reduced NAD is written as NADH plus this hydrogen ion. NADH delivers the electron pair to the chain, and NADH becomes NAD with a positive charge. With each redox reaction, the electrons lose some energy, which is used to pump protons 2 at a time across the membrane. Eventually, the chain transfers the electrons to a final electron acceptor. Anaerobes typically use inorganic molecules as final acceptors. For example, some prokaryotes reduce sulfate to hydrogen sulfide. Others reduce nitrate to nitrite or to nitrogen gas. And methanogens reduce carbonate to methane. In aerobes, the final acceptor is oxygen. A pair of electrons and 2 hydrogen ions combine with half of an oxygen molecule to form a molecule of water. In summary, NADH delivers pairs of electrons to the membrane-bound electron transport chain. The electrons pass from one molecule to the next in redox reactions until they eventually arrive at a final electron acceptor. The electron's energy is used to pump 6 protons across the membrane. A different electron carrier, FADH2, comes from the Krebs cycle and delivers another pair of electrons, but further down the chain. These electrons release only enough energy to pump 4 protons across the membrane before passing to the final electron acceptor. The result of electron transporting and proton pumping is many more hydrogen ions outside the membrane than inside, producing an electrochemical gradient across the membrane. Electrochemical gradients provide energy to drive some cellular operations. Let's consider one, the synthesis of ATP. Cells make ATP by a procedure called chemiosmosis. They do this using ATP synthase, or ATPase, which is an enzyme that uses the energy of electrochemical gradients to make ATP. In chemiosmosis, ions flow across the membrane through a channel in ATPase. As ions flow through the ATPase, parts of the molecule change shape and rotate. This mechanical motion causes the enzymatic activity to add inorganic phosphate to ADP to make ATP. To summarize, NADH and FADH2 deliver pairs of electrons that move down the chain. Each molecule in the chain is alternately reduced and then oxidized, energy is released, and protons are pumped across the membrane. An electrochemical gradient forms. The electrochemical gradient can be used to drive the synthesis of ATP by ATPase.